If you want to increase photosynthesis, then we really need to talk about iron. So today in this video, we'll be talking everything you need to know about iron, managing iron in your soil, in your plants, and how that translates into production. So super interesting video, and iron is the first, at least in this series, that we're running, the Regenerative Nutrition Management series of our trace minerals. So we're getting into redox, which is super interesting and complex, which is how the iron interacts in our soil. But anyways, to start off with iron. Iron, super important for, as a general concept, photosynthesis. There's 3 minerals for photosynthesis, well there's 4. There's nitrogen, and we probably have too much nitrogen as it is. What we're really lacking in is likely the other 3, which is magnesium, manganese, and iron. So iron specifically is required for the synthesis, so the putting together of chlorophyll molecules. Chlorophyll molecules are the things within the chloroplast that actually capture the light that come into the plant. So without the chlorophyll, you can't capture the light for photosynthesis. So we talked about in the magnesium video that magnesium sits at the center of that. It's the iron that actually puts it together. So without iron, you don't get the construction of chlorophyll. Likewise, without magnesium, you don't actually get the complete structure, even with iron. So you need iron, magnesium, and manganese. We'll get to that in a future video on manganese, but super important that you have iron. So that means if your plant's sitting out of bricks of say 3 to 6, even like 9, potentially iron could be a reason why that's not higher. Ideally, we want to be aiming around that 12 bricks to start getting the benefits of increased photosynthesis, such as increased tolerance to pest and disease. And so iron is critical for that. Now, on a very similar token, iron is required in the synthesis of carotenoids. Now, carotenoids, similar to chlorophyll, can capture light and then pass it to your chlorophyll. Now, this is important because what you'll see here is the light captured or absorbed by our chlorophyll A and B, as well as our carotenoids. So you can see here in the full line of green, chlorophyll A captures light at that 450 mark, dips down as it goes green, and then captures more light in the red zone. Chlorophyll B is offset a little bit, so it's capturing more light towards the green. But then, again, it dips down, little bump here, but otherwise that's it. What carotenoids do, and this is why they're quite important, and another reason why it's important that you have iron to produce them, is that it increases the photosynthetic ability further into the green spectrum, which the plant typically isn't capturing, as it is just with the chlorophyll A and B. So you can see here, it comes across a bit, so it's capturing more light in this zone, but additionally across the spectrum of chlorophyll B. So not only does it just increase overall photosynthetic ability, but also increases the ability of the plant to photosynthesize in using light, which is more towards that green component of the spectrum. So both of these are required for photosynthesis and capturing that light, and it's iron that actually allows for the synthesis of these. So just to drive home the point, in our book of mineral nutrition in plants, there's a few studies and references talking about the effect of iron deficiencies and having sufficient iron on chlorophyll content. So in the first one, you'll see here talking about iron concentration within the leaf and chlorophyll concentration in the leaf. Pretty much the highest concentration had the highest amount of chlorophyll, and it's pretty much a linear response. As you decrease your iron, you decrease your chlorophyll. So it got to a point where it was less than half of the amount of iron concentration led to half the amount of chlorophyll. You could almost effectively assume you're going to double your photosynthetic ability if you have a deficiency going to a sufficiency with iron. So that's pretty powerful. Skipping ahead a little bit, ferredoxin, which is that one, also decreases with decrease in iron concentration. We'll talk about that just in a moment. Another relationship between iron supply, chlorophyll content, and leaves. Iron sufficient plants have a high concentration relative to iron deficient. And then finally, we're talking about iron supply, chlorophyll A and B, as well as total carotenoids here, but also rubisco speed. So there's a important relationship with iron and rubisco. I think it's in the formation and synthesis of that, where it takes the carbon dioxide and attaches it to different compounds to then produce the sugars. So you can see chlorophyll A significantly decreases with iron deficiencies, likewise with total carotenoids. So we're going from, say, 400 micromoles to, say, 30 in extreme iron deficiency. So a 10x reduction in your chlorophyll A content. Likewise with the total carotenoids, you're almost getting a 10 times reduction in that too. So pretty important that we have iron supplied to our plants so that they can photosynthesize at their maximum rate. And as farmers, that's really our job, is to allow the plants to photosynthesize so they can produce photosynthetates or sugars to then feed whatever we want to produce. So whether that's pasture, we need to increase the biomass of the pasture, we need carbon to do that. Or if we're trying to produce grain or fruit, we want that carbon to be pumped into that grain or fruit. So we need to make sure we're maximizing the photosynthetic ability of the plant, and paying attention to iron is critical to doing that. Now, another very important function of iron is in the development of iron sulfur compounds. So an example of that is ferredoxin. So this compound effectively takes electrons from other things and then passes them to another thing to use that electron as for another chemical reaction. And so ferredoxin is used in a whole range of different things, but there's three main ones that we need to pay attention to. The first one is photosynthesis. So during photosynthesis, there's a part where electrons need to be transferred from within the cell to outside the cell. What ferredoxin allows for is that electron to pass through, or ferredoxin holds that electron, pass it to another compound to then produce energy for the rest of photosynthesis. So it's a very small part, but it's critical. And it's super important that iron is there to allow for effectively this recharging event using electrons. We're talking about the importance of iron and electrons in its redox states in just a moment. But basically this allows for electrons to move around in the plant. So that's in photosynthesis, a very similar one in respiration. And the next one is nitrite reductase, so once nitrate converts into nitrite, that nitrite then gets converted into ammonium. So iron and sulfur with this ferredoxin is required for the nitrite reductase to work. So potentially if you don't have iron, you can get a buildup of nitrite, which is super toxic to the plant. It's not great. So we'll make sure we've got plenty of iron for that. Finally, leg hemoglobin. So this is similar to hemoglobin in our own blood. It carries around oxygen. Legumes specifically need that for keeping oxygen in low concentration in the nodules. And so it needs iron in this structure to then transport oxygen in or out, just buffering that oxygen environment. So overall, we have these three really driving photosynthesis, a little bit on nitrite and respiration, and legumes. So super important for all plants, especially legumes, super important. Now, one of the problems with iron is that in its free form within the plant, it produces radical oxygen species. What these do are effectively really damage the plant from inside. So a lot of the times iron is held up in the cell structure within cells and cell walls, which means it's a non-mobile nutrient. So it's held up in the plant. It doesn't really like to be transported within the plant, which means if you're going to get a deficiency, that deficiency is going to be in the youngest leaf if supply stops from the soil. So that means when we're taking our differential sap tests, and if you do have a differential sap test, you can actually use our free differential sap test analyzer. It's on our website. Basically what happens is you take your old leaf and your new leaf, so two separate samples, send it to the lab, you get some numbers back, you put into our tool, and I'll analyze that for you. But effectively what we're looking at is the difference between the young and the old and the young leaf. With iron, if there's more iron in the old leaf and less in the young leaf, it means that for some reason, iron is not moving at the same rate into the younger leaves as it was before. That could suggest a deficiency. So typically we're looking at a 10% variation. Outside of that, once we get past 10%, and then we're starting to go, okay, is this a deficiency? Do we need to apply iron? I talked about iron as a carrier of electrons. And what this means is that iron is able to hang on to electrons and then exchange that as well. So it's able to catch the electron and throw it away. What this means is that iron typically exists in two oxidation states, iron 2 and iron 3. Now this is both important for the function of iron in these particular roles, as well as the transport and uptake of iron and the availability of iron in the soil. The only difference is that iron 2, which is the reduced form, has more electrons, and iron 3, which is the oxidized, has less electrons. So if iron 3 gains an electron, it turns into iron 2, and likewise if iron 2 loses an electron, it turns into iron 3. Super important you understand that with iron because it drives a lot of its functions. So then when it comes to the iron in our soil, there's so much iron in our soil that we usually never actually have to apply it. The only exception to that is in very sandy soils where you just don't have that much iron. A lot of it's just silicates. But if you have really any clay content, you'll have iron in your soil. So we can look at the total iron in our soil tests. If you don't have one of those, that's basically the base of our nutrition program or the programs that we work our clients through. Basically, we want to know exactly what's in our soil that could be available to our plants, say in 1,000 years, which is what the total soil test does. Iron is the fourth most abundant nutrient in our soil. So after oxygen, silica, and aluminium, iron's next. So it's very, very unlikely that your soil is actually iron deficient. What usually is the problem is that there's a availability problem with your iron. So that means that there's not enough soluble iron 2 that is, in the soil. So I mentioned before, the oxidation state is important for uptake. The plant can only take up iron 2 as it is. In order for the plant to take up iron 3, it requires the plant, as well as microbes, to put out what's called cytophores. What these do is a chelating organic compound which holds the iron in a particular way that allows for the plant to then take them up. So once the iron's in the plant, it would rather transport iron 2 throughout the plant. In iron 3, the plant can't actually quite use it, which is also why it's super important to get a SAP test rather than, say, a tissue test, because a tissue test doesn't differentiate between an iron 2 and an iron 3. A SAP test does. So if it's in the iron 3, it can go all through the plant, but the plant can't actually use it unless it converts it into iron 2. So ideally, we want to have everything in iron 2, so it can be taken up easily, transported through the plant, and then actually used in these things. Because there's no point in having iron in the plant if it's not actually used for anything. So what that means is we need soluble iron in the iron 2 oxidation state. Iron 2 tends to be the most soluble, which means there's not too much of a problem with iron 3 unless the soil is oxidized. So again, there's two states, there's oxidation or oxidized, or there's reduced environments. When we're talking about the reduced environment, which is more favorable for iron uptake, we need to think of, say, waterlogged conditions or like sour conditions. That is typically reduced environment. So waterlogging and lack of oxygen, anaerobic. Aerobic and highly oxygenated areas causes oxidation, which is the iron losing electrons. So we want to make sure the iron has lots of electrons. Best way is with waterlogged conditions or, and we'll talk about this in a moment, well-structured soil. So typically, soluble iron is really dependent on the oxidation state of the soil, and that typically depends on how we manage our soil. So iron 2 availability increases with both reduction or reduced environments, as well as with acidity. So you don't want too much acidity. So around that 6.5 is the best. If you start going down into, say, 4, iron can become toxic that we don't want. It's going to antagonize a lot of other things. Likewise, if it's too reduced, that's also going to create iron toxicity. So you want a nice, slightly acidic, slightly reduced state in our soil to optimize our iron availability. Typically, our agricultural soils are oxidized, meaning that through tillage or herbicide applications, and glyphosate is a very good oxidizer of minerals, that a lot of our iron is going to be in the iron 3 phase, which is not easily available to the plant for uptake, which means a lot of our plants in agriculture could actually be iron deficient despite having so much iron in our soil. And so there's a few main situations where iron deficiencies are quite common. The first one is in sandy soils, which is a problem here. We don't actually have the iron in the soil to actually give to our plant. So that's the first problem if you're in sandy soils. We'll talk about what fertiliser add just in a moment if that's the case, but typically that's not the case. But getting a really good soil test done will determine whether or not that needs to happen. Typically, it won't, which means the next reason why you could have iron deficiency is you have a high pH or high redox, or a oxidized soil, high redox. So both of these things will reduce the iron 2 availability. The pH will drive it towards insoluble forms, whereas the oxidation state will drive it into an oxidized state, which means it's not available in the iron 3 phase. Finally, the last one is high calcium and high manganese levels, as well as I think copper and zinc will also antagonize iron. Effectively, that's more of an antagonism with other things. Where this comes into place is after an application of lime. Lime is very oxidizing to the soil environment, which causes iron 2 to go to iron 3. Likewise, it supplies the calcium that can antagonize the uptake of iron. So typically you might get a reduction in iron content after a lime application, especially if it's very high application, more than 4 tons a hectare. They can really reduce iron availability. So to recap, total iron is usually not the problem unless you're in a really sandy soil. The problem is with availability, which is typically a problem with the redox and pH. Cool, so now that you understand the overall idea of this, for total iron, what we're looking at is a total iron of 1,000 parts per million. So if we have more than 1,000 parts per million in our total iron, then we don't have to worry too much about the total iron being a problem. Now, it could be less than that, and it probably won't be a problem, but it's at that point where you can start considering, okay, if we don't have available iron, is it because the lack of availability, or is it because lack of actual iron? And so that depends. But really, it's at 1,000 parts per million, which is kind of that trigger point or threshold to then consider starting to apply iron. For our soluble, we're looking at how much? 15 parts per million. If you have some soil tests, CEC style soil tests, you can actually get this free report using your current soil tests on our website. Likewise with the free differential sap test analysis, just go to our website, plug in your numbers, and it'll spit out a free report with the ideal levels to add everything to. So both nutritional recommendations and ideal levels and a nice formatted report. But anyways, for our available iron, we're looking at about 15 parts per million. Now, the problem with this is that I don't think this will differentiate between iron 3 and iron 2. So that's one of the problems with the soil test. A SAP test will tell us whether it's in the iron 2 or iron 3. The other problem is that this can change within hours. And so you might have a oxidized soil one day, then you get a shower of rain, it gets slightly waterlogged, or it reaches fill capacity. Then everything turns over into iron 2. So redox changes quite quickly, whereas pH on the other hand doesn't really change, which means you can take that number as a pretty reliable indicator of availability if it was a reduced state. But again, some of our soils don't really get to that reduced state because of our cultural practices. And so for the available iron, anywhere between 15 to 25 for a heavy clay. So a heavier clay will require more parts per million than a sandy soil. So sandy soil, heavy clay, in that range is somewhere pretty good. But again, the key thing to tell us whether or not we have a iron deficiency is a differential SAP test analysis between our young and old, and we're looking at a 10% variation to inform us whether or not we're starting to go into a deficiency. Typically, if that's the case, then we look at all of this data and go, okay, there's a deficiency here. Is that because of the available iron? Is that because of the total iron? What's the go here? You need all these pieces of the puzzle to actually tell us what's going on. Cool, so let's just say we look at our soil tests, we have a sandy soil, and we go, okay, we only have 300 parts per million of iron in our soil. So from here we go, okay, we actually need to apply iron because we just don't have enough iron in the total component of our soil to actually give our plant iron. What we need to do is a soil application of iron. Rarely do we do this, but and get iron granules that you just broadcast out. So you can work out your application rate, do it across multiple years, but it's just an iron application, iron granules. It's pretty straightforward. If, for example, we have sufficient amount of iron in our total, we have sufficient amount of soluble iron in our soil test, but we're just not getting it in our plant. The question is, okay, do we apply something? And if the answer is yes, what are we going to apply? So typically with foliar applications, and especially in this system where there's plenty of iron in our soil, it's just not getting into the plant. We want to make sure that the plant is still photosynthesizing effectively, and so we need to supply iron. The role that foliar applications have is they just give the plant that little bit of kick to then kickstart the system. And so the overall aim of a regenerative approach is not to get the plant hooked onto foliar applications. It is to use foliar applications as a way to kickstart the system to then get the plant to produce more root exudates, to then feed more microbes, to then get the microbes to supply the iron. So that might occur over multiple years, but every time you make a foliar application, the plant should be getting stronger and stronger to then improve the soil biology. So for foliar applications, if we look at our differential sap test and it goes, okay, we're iron deficient, there's plenty in the soil, so it's not a soil problem, it's just an availability problem, and we're going to apply it because we're going to be losing yield because we're not getting efficient photosynthesis. What are we going to apply? There's really two main forms. There's a few others, but really you got iron sulfate, super good enough, and iron chelates, also super good enough. Both of which are fine. I don't think there's too much of a preference. There is a bit of a preference, but I don't think there's too much. It's not that important. If you're going to use iron sulfate, make sure to add fulvic acid because it'll chelate it and increase the availability. There could be a problem here. So this is currently in the reduced form. There could be a problem if you apply that and it oxidizes on the leaf. So that could be a problem. You just have to, if you're going to make your own foliar applications with iron sulfate, you probably just need to experiment a bit, even do your own research and development and take a differential sap test beforehand and after the application to see if the iron is getting into the leaf or if it's oxidizing. The fulvic acid should be able to chelate that and get into the plant. If not, the iron chelates should be able to do that. And there's a whole range of different things that they use to chelate it. Some organic compounds such as amino acids, other inorganic compounds like EDTA, I think. Either one, I don't think it really matters too much. The end goal is to apply your foliar applications so that the plant can photosynthesize more to then feed the biology, for the biology to then feed the plant with iron, so you don't have to continuously add iron. So when we talk about regenerative management, what we want to do is optimize our plant performance with nutrition and biology, with the end goal to reduce unnecessary nutrition inputs. So for example, their iron, there's actually plenty of iron there. So from a regenerative point of view, we don't really want to be increasing or making iron applications because it's cost money. So if we can get our soil and soil biology to supply iron, that's really good. But we need to get to that point first. So for our regenerative management, the very first thing we needed to do is a total soil test and make sure that our soil actually has iron in it. So take a soil test, a total soil test, making sure there's more than 1,000 parts per million. What we then do is check the availability. Is it available to the plant? Let's just say we don't have a total iron. We then add our iron granules. If not, and that's all right, we take a differential sap test throughout the season. If that's a problem, we add our iron foliar as a quick fix. These are fixes, they're band-aids. What we want to do is eventually get to a point where we're improving the soil. The biggest component of this is reducing our soil environment. When we talk about that, there's kind of two things going on. We don't want a waterlogged or compact soil, which is also reduced. What we want is a well-aggregated soil, which has both areas of reduced and oxidized stages. So the only way to do that is with well-aggregated soil. So the thing about aggregation, water and gas or air can flow in between our aggregates, which is what we want. We want a well-aggregated soil to depth. The outside of this is where the oxygen is, where our roots can go, where our water can get in. That's what we want. Inside our aggregates is what effectively we want is a reduced environment, which is where the plants and biology can tap into our soluble iron. So if we have iron 2 in our aggregates, the aim of the game then is to how can we increase our soil aggregation and soil structure. This is how you build a long-term supply of iron. Now the other thing with iron is that you need a continuous supply of iron so you don't get deficiency. If, for example, you have an oxidized soil, a sandy soil, and it oxidizes quite easily, the iron will be available in sudden bursts. As the soil gets wet, it will then release a lot of reduced iron. As it dries up, it will oxidize, and then you'll get, the tap will turn off for iron, which means your younger leaves will become iron deficient. And the younger leaves are super important for photosynthesis. They're the most photosynthetic leaf of the plant. So we want to make sure our plants get a continuous supply of iron, which means the soil needs to contain sufficient amount of iron 2, which means there needs to be little pockets of reduced. So the question again is, how do you increase soil aggregation? The way to increase aggregation is with healthy plants. So healthy plants release root exudates to feed biology. The biology then stick particles or soil particles together to create aggregates. So in a way, we need to then supply our iron foliar to then increase our plant health, to increase the photosynthetic ability of the plant, to then increase the amount of root exudates, to then increase the amount of aggregation, to then increase the amount of iron supplied back to the plant, so that we don't have to apply more iron foliars. So really, the way to increase aggregation is to increase the amount of root exudates that the soil receives. There's 2 ways of doing that. There's the rate per unit of time, and then there's the amount of time. So the first one of that, increase the photosynthetic ability per unit of time. That is the efficiency of photosynthesis. That is where the iron applications come in. If we only have enough rainfall for one crop, we can't put a cover crop in, we just have a 1 crop of wheat each year, or annual species per year, then we need to increase the photosynthetic ability of our plants to then 4x, so the photosynthetic ability, to then 4x root exudates. In a system where we can have, say, a pasture phase, or we can have a cover crop, that's going to extend the unit of time, or the time period that we can photosynthesize, which then, even though the photosynthetic ability or the root exudates per unit of time is lower, say if you don't have iron application, you have 4 times as long, so you get the same result in the end. So really, the way to increase aggregation is with increased plant health, which comes back to foliars. Now there's a few other cultural things like tillage. Tillage destroys aggregation, and so we want to really go no-till or have a reduced till setup. Likewise, herbicides also creates an oxidized environment, so we don't want to be doing too much of that. So it's a tricky one. Do you till or do you apply herbicide? It really depends on your operation, what you want to do. Either one will oxidize the soil. So it's how do you do it so that we're going to create the least amount of damage possible. A light disking seems to be okay. And likewise, a herbicide application is just quick and easy. Now, the other way we can increase aggregation is with the application of, say, a soil primer. We have a whole video on soil primers, so you can go check that out. Effectively, what we want to do is apply a bit of labile carbon. So a really good source of that is humic acid or humates. They're a complex carbohydrate. You can apply it to the soil, micro eat it. Fungi love it, and they increase aggregation. But the thing is, you can't replace the root exudates from plants with a soil primer or molasses or fulvic humic acid. And so to put that into perspective, so on the assumption that a plant pumps as much carbon into the soil as it does into its biomass or above ground biomass, a cover crop or a crop at, say, 10 tonnes of dry matter per hectare will produce 10 tonnes of root exudates per hectare. Compare that to a soil primer where you're putting 10 litres of molasses on per hectare. So 10 tonne versus 10 litre, root exudates are the cheapest way to get sugars into your soil to feed the biology. Soil primers are cool. They only really work on the top bit. You need the rainfall, you need all these things. You don't get the bulk. So a soil primer can help, but really there's no substitute for growing really healthy plants. There's a few different plants that are really good at causing aggregation and reducing the soil environment. A really good one is oats. So adding oats to your rotation is a good one, as well as sunflowers is another really good reducer. But really, growing any plant, especially a diverse cover crop, for example, you have lots of different roots, different depths, different ecological niches are being used, maximize the overall root exudation. So we can get a bit idealistic about, we need to make sure we have the best plant available, but really any plant will do. The more diverse, the better. But even just having one crop of whatever in will be better than having nothing. So there you go. That's how we manage iron regeneratively with our clients. Really, it's looking at totals available, whether or not it's in the plant, oxidation states, and then trying to fix our plants with a foliar application. And yes, it's Band-Aid fixes, but the idea is that the plant increases its photosynthetic ability, feed the biology, create a bit of aggregation to then supply the plant with iron into the future. So if you want to implement some strategies like this, you can sign up for a free consult. Sit down with me for 30 minutes. We'll go over 5 recommendations for your farm, implementing stuff like this. Otherwise, you can work with us through our consulting programs. You can head over to our website, check it out. Awesome. Thanks so much for watching. My name's Till. Cheers.